Hi, today I'm presenting my paper on RepNet for quantifying the reproducibility of graph neural networks in multiview brain connectivity biomarker discovery. This work has been done at Basira Lab by myself and supervised by Dr. Islam Rekik and Professor Serdar Çelebi. Graph neural networks, GNNs for short, have become crucial as they have the capability to learn graph representation of brain connectivity while preserving its inherent topology. Nevertheless, concerns have been raised by researchers about the reproducibility of models in these applications. Reproducibility in this context refers to the ability to consistently obtain the same top biomarkers when the model is trained with different connectivity measures and employs various hyperparameters. There are some studies when it comes to reproducibility on various methods for multigraph connectomic datasets. For example, one introduced a preliminary approach aimed at identifying the most consistently reproducible connectomic features when employing various feature selection methods. However, this approach relies on basic feature selection techniques and isn't well suited for assessing the reproducibility of GNNs. On the other hand, another work presented a framework based on graph topology to select the most reproducible GNN model from a multigraph connectomic dataset that encompasses different views. Although this approach assesses the reproducibility of GNN models, it doesn't account for the interview reproducibility of GNNs. To overcome these limitations, RG-Select proposed a framework that combines GNN to GNN and view-to-view matrices, varying the number of top biomarkers. However, existing methods have issues such as information loss due to matrix averaging operation, lack of validation for biomarker consistency, and quantifying interview and intermodal reproducibilities with the same method. To address these, we introduced RepNet, a primary method assessing GNN model reproducibility using interview and intermodal reproducibility scores. Here we explain RepNet. First, the dataset is divided by the different views. These datasets are further split using an k-fold cross-validation to vary the data distributions. Then, GNN models are trained using the datasets. Second, the features from the final layer of each GNN model are extracted and normalized across all features. Normalizing and taking the absolute value of a biomarker vector helps determine how each biomarker influences the classification outcome, allowing for the quantification of their impact. Third, view-based matrices are formed by concatenating features within each view horizontally. Biomarker vectors are grouped by view, regardless of the cross-validation fault, and view-based vectors are derived by calculating their L2 norm row-wise. Finally, the distance vector d sub m is computed by calculating the squared L2 norm for each combination of view-based vectors. The interview reproducibility score for the relevant GNN model is determined by measuring the Euclidean distance of this vector from the origin. We tested our methods against RG-Select using a dataset include 150 autism spectrum disorder patients and 150 normal controls. Multigraphs for the right hemisphere and left hemisphere were created using 6 cortical measures. In our experiments, we employed 5 different GNN architectures, namely diff pool, GAT, GCN, SAG pool, and graph unit to diagnose whether a brain graph indicated autism. Our experiments focused on assessing interview reproducibility, investigating reproducibility through interquartile range analysis, evaluating intermodal reproducibility, and identifying the most reproducible biomarkers. We compared RepNet and RG-Select to assess their interview reproducibility scores. Despite different scoring methods, we evaluated them based on how they ranked GNNs in terms of reproducibility. We expected the internally consistent method to maintain the same ranking, regardless of the training dataset or the number of top biomarkers chosen. Remarkably, RepNet consistently provided more robust rankings than RG-Select. RepNet employs the L2 norm, which highlights differences between biomarkers, revealing true reproducibility capabilities. GCN emerged as the most reproducible model, with GCN and diffpool standing out as significantly better performers, closely matched in reproducibility. We introduced a third evaluation method separate from RepNet and RG-Select to ensure a comprehensive comparison. By grouping biomarkers ranks from diverse training datasets and calculating interquartile ranges, we measured how rankings changed with varying data. Our model rankings, with the most reproducible at the bottom, shoved biomarkers closer to IQR 0 indicate better reproducibility. Notably, GCN outperformed DIFPool with consistently lower IQR scores across all biomarkers. Both GCN and DIFPool surpassed the other models, demonstrating higher reproducibility. We found a significant quantitative difference reproducibility, aligning with RepNet's method and reinforcing RepNet's superiority over our G-Select. To calculate intermodal reproducibility, we average the index vectors from each view for each GNN sub-n. This forms a GNN to GNN matrix that measures how many normalized top biomarkers are shared between models. The result is a list of intermodal reproducibility scores for each GNN. Notably, GNN emerges as the most reproducible model, although its scores are lower than those in the interview analysis. This may be due to an assumption made during intermodal reproducibility calculation, implying that most reproducible model shares all its biomarkers with others. However, GCN's strong performance in both interview and intermodal reproducibility underscores its reliability in distinguishing between different brain states. We identified the top 3 biomarkers from the GCN model to assess their potential in distinguishing between autistic and normal brain graphs. These biomarkers include the precuneus cortex, the bank of the superior temporal sulcus, and the lateral orbital frontal cortex. Our findings align with the existing research, as one study revealed that individuals with autism exhibit weaker functional connections between the precuneus cortex and various brain graph regions. Furthermore, another study by Gervis et al. Demonstrated reduced activation in the superior temporal sulcus among autistic individuals. Finally, McAllen et al. Reported that individuals with autism exhibit lower gray matter in the orbital cortex. This study introduces RepNet, a novel approach for quantifying GNN model reproducibility in the context of consistent biomarker discovery. RepNet differentiates between interview and intermodal reproducibility, ranking various GNN models based on the consistency of their biomarkers across different views. Our research demonstrates that RepNet outperforms existing methods, achieving results that are both internally and externally consistent. In our future work, we plan to validate RepNet's findings using extensive connectomic datasets. Thank you.